Today we have with us Nobel Laureate Dr. Jack Szostak. Dr. Szostak was part of the trio who received the Nobel Prize for Physiology or Medicine in the year 2009. He is professor of chemistry at the University of Chicago in the US and advisor to Trivedi School of Biosciences at Ashoka University in Sonipat, Haryana. Thank you very much for speaking with PTI. It's an absolute pleasure to have you with us.
Oh, thank you for having me here.
So before we discuss the discovery that you're most recognized for, let's briefly discuss what are telomeres, right? Could you please just briefly explain what are telomeres?
Yeah, sure. So telomeres are special sequences found at the ends of chromosomes, which are very long, near DNA molecules. And they're wrapped up with a whole bunch of proteins, and they play an important role in keeping chromosomes intact. And it turns out they also are important for things like cancer and aging.
Exactly. So we were going to come to that. And given how telomeres have been found to be important for cell aging processes, how would you describe for an average person the methods or the ways through which one's actions, daily, everyday actions, could impact health of telomeres?
Well, so it's not something I've worked on personally. I mean, I worked on the basic biochemistry of this a long, long time ago.
Yeah,
I don't work on it anymore.
I see.
But I follow this field from a distance. My understanding currently is that the telomeres, their regulation, their control in biology is very complicated, not that well understood. But some things like a lot of stress apparently have effects on telomeres, which leads to them getting shorter with time, which will increase cell death in tissues, stem cells. And so it might be a contributing factor to some of the problems with aging.
Would you be willing to briefly explain how the discovery of the protective role of telomeres happened?
Yeah, sure. So back, let's see, almost, 45 years ago, I think I met Elizabeth Blackburn at a scientific conference.
Okay.
She gave a fantastic talk about telomeres in a kind of bizarre organism called tetrahymena. It's something you find in swimming around in ponds in the summer. And she was the first person to work out the sequences of this DNA at the ends of chromosomes in tetrahymena. I went up to talk to her, and we had a great conversation and agreed to do a collaboration. And at the time, I was working on yeast genetics, and so I thought we could look at telomeres in yeast. See if they looked similar, maybe learn something about how they work. And so that went very well. We were able to clone yeast telomeres and find that they were similar in some ways to the telomeres from tetrahymena that Liz had studied earlier, but also had some differences. And as our work progressed, that led to the hypothesis, the idea that there might be an enzyme, now called telomerase, that adds DNA to telomeres, so it makes them longer. And this counteracts the inevitable shortening of telomeres that happens during repeated cell divisions. And so there's been so much work on it ever since. A lot focused on the enzyme telomerase that adds extra DNA, but also on the reasons for why telomeres get shorter over time. And it's become a really active area of work. I think there probably thousands of people or labs working on this subject, trying to understand exactly what controls telomere biology.
I was actually going to come to that point. Uh, how do you perceive the importance of your discovery now, after all these years, when aging and longevity, and telomeres have been studied to have profound importance towards aging and longevity. So and they are hotly discussed, debated topics. So how do you perceive the importance of your discovery now?
Well, I think the real importance so far is that it has really opened up this area of inquiry. There haven't been any magic solutions to the problems of aging or cancer, but we recognize now that the control of telomeres is an important part of both processes. I think one thing that makes it really complicated is that if you did something that would just increase telomerase activity so that telomeres are always getting longer in all cells, that would probably lead to more cancer. And conversely, if you inhibit telomerase in all cells, like if you're trying to treat cancer, that would probably make aging problems worse. So it's this, what makes it complicated is this balance between effects on cancer and aging.
Right. So, what is that sweet spot then?
Well, I think if we really want to have a beneficial effect on some aspects of aging, one thing that might be helpful is if we knew how to control telomere length, in other words, the activity of this enzyme telomerase, at the right time in the right place, right? In the right cells, not in all cells, just in the cells that we want to help. But that's very difficult because we still don't understand fully the way that the enzyme is regulated.
Right. Okay. So we have large genomics projects that are being carried around the world, and they have been carried out since the team has made the discovery that earned the Nobel Prize. So the Human Genome Project is a landmark one, where uh, where where a human genome was generated for the first time. Then several genomics projects and genome-wide association studies are underway around the world, including the Genome India Project, right? What is the nature of knowledge that we gain or we stand to gain from analyzing genes and genomes on such a large scale?
Wow, that's hard to answer because there are so many answers, right?
Okay.
But understanding the whole human genome at a population level, so thousands, now millions of people have had their genome sequenced.
Yes.
And so it gives us a huge amount of information about the causes, the genetic causes of diseases, many, many different diseases, resistance and susceptibility to different diseases. It gives us a huge amount of information about the migration of populations of people over time. So it tells us about the history of humanity in that way. And because of new technologies like the use of CRISPR to manipulate the genome, we now have the really interesting but somewhat scary prospect of actually editing the genome, where the good things would be to maybe cure some genetic diseases, and we're seeing the first examples of that already. But then it's obviously very worrisome if changing our genes changes the nature of humanity over time.
In what ways does the knowledge from genes and genomes further our understanding of human biology?
Well, I think it furthers our understanding, again, in a lot of different ways. So, how does an adult human develop from a fertilized embryo? I mean, there's so many different things happening there as a cell divides and differentiates into different kinds of cells and tissues, organs. And so a lot of understanding that will come from knowledge of the sequence of the genome. Of course, a lot of those studies in developmental biology are done in model organisms. And again, the gene sequences are critical. But that's only a starting point, right? Knowing the sequence by itself isn't that helpful. You have to know how to interpret the sequence, in terms of what RNAs or proteins are coded, what they all do, how they interact with each other. So, if you were just given a book that gives you the sequence of 3 billion letters, it's unreadable, right? You can't get anything out of it just by staring at the sequence. So you need to take in all the other tools of molecular biology to try to understand how a genome actually works in the context of the cell that it's in.
I'd like to ask some India-related questions.
Sure.
Yeah. The Genome India Project is an initiative to put forth and maintain a database that is representative of Indians. Whole genome sequencing of 10,000 individuals in India has been done. A preliminary set of findings have been published, but detailed analysis are underway. Databases around the world are largely biased towards those with a European ancestry, and this has been repeated quite often.
Except I understand there are some very large genome sequencing projects going on in different places in Southeast Asia. I think that Singapore is leading one such initiative with a goal of 1 million genome sequences. And I think in China also very large-scale sequencing projects are underway. So, yeah.
So around that, how would you describe the importance of India's genetic database towards global research, and in what ways does it contribute, do you think?
Probably the most easy to talk about aspect is simply for medical research, understanding genetic diseases, identifying the most common genetic diseases, ways to either identify them very early on or treat them with some of the new molecular technologies. So I think that's driving a lot of it. But the other part of it that's really interesting is also how the population has emerged or evolved over time. Obviously, within India, there have been many, many large-scale migrations over history, and understanding the history of the country through this genetic tool is just one way of helping to understand the history of the population.
When we talk about health and disease, it has been said very often that Indians, among other ethnicities in the world, are genetically predisposed to disease. Very often repeated in the context of chronic, non-communicable diseases such as, and not limited to, diabetes, heart disease, hypertension, among several others. So keeping all other factors aside and looking only at the genetics aspect, is there anything that can be done about the genetic risk, the genetic propensity for diseases?
I don't think there's any easy way to manipulate or even desirable way to manipulate the genetics of a population, right? That would be a really a bad way to go. but the hope is that the understanding of how all of these diseases emerge from the interaction of the genetics with the environment, that will teach us something and teach us about how to minimize and avoid these kinds of diseases. They're not just genetic causes, right? They're largely the interaction. There may be genetic aspects that increase susceptibility, but it's a susceptibility to the environment, the way people eat, the way they exercise or don't. It's things like that. And if we understand that, we can advise people on how to live a healthier life, and we can also develop therapies for when problems arise.
In your understanding, what is the larger trajectory that genetics wise a population takes before achieving optimal population health? How does the story unravel to you? Does that make
I don't
sense?
really understand
Okay.
the question.
What I'm trying to ask is that if there is a if a population has a genetic risk, let's say towards one disease, all right. So how do and genes or rather a DNA sequence is mutated when
Okay. So like in
through
a disease
inheritance.
with a single gene cause.
Yeah. So like through inheritance,
Mhm.
the sequence changes. So would you have any idea as to how a population or like genetically it the sequences change before they make way for
Well, okay. Well, there is a well-known phenomenon um, in which there are certain genetic diseases that are caused when a particular gene is inactivated by a mutation on both the mother's and the father's side. Right? So the child has two mutant copies of a gene and will then be ill and will experience this genetic disease.
Mm-hmm.
And sometimes these, you would think that would be eliminated by natural selection, right? But one way that that such diseases are not eliminated, in fact can increase, is if there's an advantage when an individual has one mutant copy and one normal copy. And so there are several well-known cases where that situation actually has an advantage. Like it may actually give you resistance to something like, for example, malaria or tuberculosis or other diseases. And so there's this unfortunate side effect that then the people who have now two mutant versions of the gene end up being really sick. And so we have that understanding. What does it let us do? I mean, one thing that some people do, not everybody thinks this is the right way to go, but since we can sequence people and since we can do very simple tests to identify such mutant genes, if two parents have different copies, right, we can say, okay, well, maybe you should either not have children because of this risk, or if you do, you can test the embryo at an early stage and have an abortion if the child is going to be sick, and just try again, hoping to get a child who has only one mutant copy. So at this stage, those are the kind of practical approaches. Maybe in the future, treating such genetic diseases will be easier, but right now it's really complicated and incredibly expensive.
Right. And yeah, so that's another question. genetic tests, genetic analysis, and getting a for an average person,
Yeah.
getting a genetic understanding of what are their risks, etc., and getting that knowledge is it comes at a quite high price. Um, how do you see this unravel in the years to come in terms of becoming more accessible
Yeah.
for the average person?
Well, if you're talking, so there are costs at different levels, right? There's just the monetary cost of having your genome sequenced. So that's just come down so much. it's unbelievable, and the cost of doing a genome sequence is becoming less and less all the time as newer, faster, better, cheaper methods for genome sequencing are being developed. So I can see in the not too distant future that it will be standard for everybody to have their genome sequenced. And then an analysis of your genome will tell you what the risks are in terms of behavior, in terms of what your children might have or not have. And that will give people the possibility to plan and decide how they want to act on that information.
You are in New Delhi at a time when pollution reaches the year's peak levels.
Yes, I've noticed.
We'd like to get a genetics perspective on the health hazard. Would you be willing to share some insights on how pollution can impact one's genes and specifically one's telomeres?
Actually, I have no idea.
Okay.
Not something I've ever, really looked at or even thought about very much.
Okay, no problem. A couple of questions on the lighter side, and involves politicians and stuff. So we have recently come across I mean it the video kind of got popular between it's a conversation between Vladimir Putin and Xi Jinping, and they talked about organ redevelopment and like things were said like if organs are continuously transplanted, people can live younger and younger, and they can live up to 150 years. So there's that chat about immortality that's going on there.
Right.
How do you see this conversation? How would you be willing to respond?
I think at this point in our medical knowledge, I think it's naive and unrealistic. The technologies for understanding organ development and transplants are developing rapidly. I mean, there is this technology of growing kind of humanized organs in these particular small pigs, which have a physiology similar to human physiology. So the possibility is there to treat some kinds of critical organ failure with transplants from animal-grown organisms. This work is, it's just something being developed now. I think it's promising, but it's obviously going to take a long time. And really, you don't want to get to the point where you're having multiple organ failure and trying to live longer by having organ transplants. That sounds like a pretty terrible way to try to live longer to me. Better if we could not have our organs degenerate in the first place.
So, Dr. Szostak, I'd like to get a view about your current engagements.
Okay.
What are the projects? What are the research projects that you're actively engaged in?
So the work that I'm doing in my lab these days, actually for the last 25 years, is about how life got started on the early Earth. So we're trying to understand how the chemistry of the early Earth gave rise to biology, the beginnings of biology.
And this is about the debate nurture versus nature, nature versus nurture. What, according to you, is that line where the genes stop and the environment factor kicks in? Or yes, so that is my question. The genes and the environment, their interaction is is what is.
Yeah, I mean, an organism is the result of the interactions between its genes and its environment. And the environment has to be thought of not just as the external environment, but also the internal environment. And the genome is obviously the product of evolutionary history. So it's a record of all of those gene environment interactions over millions and billions of years. So you can't talk about really rationally about one or the other. They're not. It's not like they're separate things that are meaningful on their own, because it's the interactions that matter.
So then, our genes are is it then just the genes? Like, if you have the genes for it, then that's it. Is that what it is, or does a human have free will, so to say?
So humans are incredibly complicated, right? Our whole physiology, our way of being, our thoughts and minds emerge, not just as a deterministic outcome of a bunch of genes that we have, the sequences that we have, right? But they're a consequence of how we've developed, how our experiences as we grew up, and our capacity to learn and to do things is a product of our experiences, not just our gene sequences.
Okay, so that brings me to the end of my questions, and thank you very much. Thank you very much, Dr. Szostak, for taking the time and speaking with PTI.
Thank you very much.
It's been a pleasure listening to you.
Thank you again.